so far we have discussed that basics of cell biology, then we came across how that microfluidics platform could be used to address that mechanosignal transductions in cell biology. Then we have discussed how to perform that different formats of cell culture in 2-dimensional and 3-dimensional in microfluidic system, and we have also discussed about the various experimental procedures should be adopted in this same platform. After that, we had gone through some important biological readouts in the cancer metastasis area, mimicking that physiological micro-confinement in the same platform, which could not be ascertained by using macro scale cell culture. Then we have discussed that organs on chip to mimic that physiology of human system, and it could be used for drug screening purposes, followed by lab on a chip model for fishing DNA genes, maybe abnormal or normal genes out of millions of gene fragments, DNA fragments, and same way to identify that proteins from that any biological sample using this microfluidic platform. And in my last lecture, means this lecture, I am going to discuss that how hybridoma technology could be adopted in microfluidic platform for the monoclonal antibody production. And before going to monoclonal antibody, we have to know about that what is antibody. So, antibodies are defensive molecules produced by the B cells in the immune system in response to foreign substance known as antigen. So, in antigen, say if you take an example, this is an antigen, it has a several epitopes, and B cells in our body recognize most of the epitopes as an antigen fragments, then B cell is differentiated to plasma cells, then it produces antibody. So, like say this antigen has a 10 epitopes, this antigen has a 6 epitopes, this antigen has a 2 epitopes. So, according to clonal selection theory, only one B cell clone will liberate only one type of antibody against one epitope. So, if we inject antigen in our body or in animal system, that our immune system will produce large number of antibodies which are polyclonal in nature. So, that is why polyclonal antibody is defined in that way. These antibodies are produced by different clones of plasma B cells against the different epitopes of an antigen. Same way monoclonal antibody, these are produced by single clones of plasma B cell against a single epitope antigens. And our immune system has a diversity to produce antibody in the range of 10 to the power 15 to 10 to the power 18 different antibodies. And here is the picture of that main class of antibody, immunoglobulin G, IgG basically. So, this molecule has a heavy chain, this is the heavy chain and this is the light chain, and this part we known as FAB part, which actually recognize that antigen epitope specifically and with high binding constant, and this part is known as FC part, means constant part. And if you are interested in immunology, you can go through that Kubi's book on immunology. I am not going to in details. Now, how to produce monoclonal antibody in vitro, because monoclonal antibody has a diverse use starting from diagnostics to modern biology lab for identification of a protein from that vast large amount of proteins using histochemistry or western blots, antibody based microarray system, and after all lot of therapeutics are coming based on that antibody. So, now how to generate that antibody? These B cells which are generated in vivo, these are not very viable, they will eventually it will die after that secreting antibody. So, if you want to produce antibody from the B cells, we have to immortalize the B cells in vitro. So, how this could be done? So, this was addressed first by Kohler and Milstein in the year of 1960s, and they developed that hybridoma technology. Basics of hybridoma technology is that fusion of B cells, antibody secreting B cells with a cancerous B cells, mean myeloma cells, then select that is selection of the fused cell, that is called hybridoma, means oma means it is a cancerous cell. Then you have to screen that positive hybridoma clones, epitope specific antibody secreting individual immortal B cell clone. So, basically for the development of hybridoma, 3 steps are there: immunization with target antigens, selective fusion, and then screening. Then out of 10 to the power 7 cells from a mice spleen, how will you select single B cell clone for that monoclonal antibody? It is a tremendous task. So, we have to adopt some selection procedure by which that only that hybrid cell, means one B cell, one myeloma cell, that fused hybridoma cells will be selected. So, how to select this? So, this is the problem, and it was discovered by that Miller in 1960s. So, that principle is that if we can block that one of the major important molecules biosynthetic pathway, like say nucleotides, then cell will not divide, cell will die. So, that nucleotides are synthesized by 2 pathways, one is de novo pathway from the precursor, and one of the important enzyme in that pathway is dihydrofolate reductase, and another pathway is salvage pathway, which utilizes that hypoxanthine and thymidine or nucleosides for that DNA synthesis. If we can block these 2 pathways, then one cell will die, but here our target is you want to die that myeloma cells, but hybridoma cells will survive. So, when we are fusing, after immunization, we take the splenocytes around say 6 into 10 to the power 7 cells, and myeloma cells around 2 into 10 to the power 6 cells, mix up, you are giving fusion mixture with the polyethylene glycol for a certain period of time, 15 minutes. Then you will be seeing that there are 5 groups of cells. This is the our required cell, hybridoma cells, along with fused spleen cells, unfused spleen cells, fused myeloma cells, and unfused myeloma cells. Our requirement is one fusion, one B cells, splenocyte B cells, activated B cells, and another your myeloma cells, and frequency of that fusion around 1% of the total cells we have used. Now, how to select this? As I had already mentioned that DNA synthesis, blocking of the DNA synthesis could be one of the best strategies for the selection. For this, hat selection media is used. What is the composition of the hat selection media? Hypoxanthine, aminopterin, and thymidine. What is the function of aminopterin? It will block that dihydrofolate reductase enzyme by which de novo synthesis is blocked, and as we are selecting that myeloma cells which are HGPRT minus or thymidine kinase minus, they have already that blocked that salvage pathway. Means in that cocktail of that fusion pot, in that mixture of hat selection media, that your motile splenic cell means your splenic B cells will die due to that 2 or 3 generation of culture, whereas HGPRT minus or TK minus immotile hat sensitive plasmacytoma will die. So, only it will survive on 2 or 3 passages or 3 generation of cell growth, that fused hybrid cell or hybridoma. This is that basic means technology was discovered around 1960s and that is still follows for the hybridoma production. So, next step is how to screen that effective hybridoma cells for the required antibody which will be monoclonal in nature. So, we have to by dilution plating that hybridoma cells will be plated in 96 oil plate by which 1 hybridoma cell will be in 1 oil. Then let it culture for 1 to 14 days according to the need, then cell will be growing, you can see under the microscope. Then you have to test that culture fluid for the antibody what we need, which will be in monoclonal in nature because these are derived for 1 B cell. So, for that we have to use indirect ELISA system. Say take for an example, this is a 96 oil plate, is coated with that antigen what we have injected in the mice, then we are giving some blocking reagent, then we are giving 100 microliter of that your culture fluid from that each oil to that antigen coated plate. Again you wash, throw it away, then you give that antibody means Fc portion against the antibody against that Fc of that antibody secreted tagged with reporter enzyme, let it bound, again you wash it, then you give some substrate for the tagged enzyme by which color will be produced, substrate to product conversion. Then you will be seeing like that. If this is the replica of 96 oil plate of this plate, then only you will be seeing that only some plates will be seeing the color, means these are the positive clone, hybridoma clone which are producing antibody. From that you have to again screen it because hybridoma cells are heterotropic fusion, these are not very stable. To make it is a stable hybridoma cell line, you have to another selection of 2 or 3 times you have to go through, by which you will be getting stable cell line and you have a stable hybridoma cell lines which will be secreting antibody. But keep in mind these are not very stable for long term. So, what are the drawbacks and challenges of this macro scale hybridoma cell culture or hybridoma generation? There is no control of fusion, very low fusion efficiency around 1 to 2%, very low yield, cell loss. How much time is required? Process time around 5 to 10 days, selection is tedious and expensive, and single cell clones cannot be selected in the initial stage. So, challenges are accuracy, cost, throughput and process automation. So, this is one of the data, means if we give 6 into 10 to the power 7 lymphocytes and 2 into 10 to the power 7 myeloma cells are allowed to fuse, only about 1% of the starting cells actually fuse, and 1 fusion in 10 to the power 5 fusion is viable hybridoma even in most efficient fusion. So, how these problems could be addressed? We think that microfluidic technology is the best technology to address all these problems, that is means fusion and screening process in high throughput way within a very short period of time, by which we can select a positive hybridoma clones for secreting one class of antibody, what we can tell is a monoclonal antibody. So, if we use microfluidic platform, what are the types or what are the problems we can address? So, first problem is that, or first step you can tell that efficient heterotropic pairing with minimal homotypic cell aggregation, means myeloma or B cell, only we want that heterotropic pairing, that is cell pairing type A and type B, this is myeloma, this is your B cell, and then selective fusion of heterotropic pairs, then next is screening. So, first we are coming cell pairing and fusion method in fluidic platform. This could be done by chemical ways, electrical field, dielectrophoretic force and microstructures, and fusion could be done by, this is chemical way, that is what we followed in macro scale, then electrical field, cell membrane, electroporation method, that could be done. first we are coming that chemical pairing and fusion method. Here principle is adopted that streptavidin avidin selection process, these are very strong ligand and receptor, there are binding constant around 10 to the power minus 15 molar. So, myeloma cells are labeled with biotin, normal chemistry is there, and B cells or you can tell splenocytes are labeled with streptavidin. Now, with the help of Y channels in fluidic platform, in one channel say B channel we are giving myeloma cells and A channel we are giving labeled B cells, then allowed to flow in Y channel, and at certain distance let it come together, and with the micromixer they will be mixing and fusion using either electrical method or PEG using polyethylene glycol. But problem is that cross reactivity, multiple biotin streptavidin interaction interfering the 1-1 interactions. What you want 1-1 pairing, it will be there, but not very high percentage. If we use dielectrophoresis method, these are the electrodes, and first we are passing one type of cell, say myeloma cells, they will be clustered around the electrodes. Then we are passing that spleen cells, activated spleen cells, and they will be clustered around that myeloma cells, and there will be pearl chain type of formation will be the cells. So, here is also that only 1-1 pair occurs, not very large extent, and then you can use pulse DC field for a few microseconds or 1 second to fuse the cells, and you off the dielectrophoresis, then you can get that fused cells, but their percentage is not very high. Due to that streptavidin avidin, because streptavidin is a 4 tetrameric complex, they also make a cluster. So, next is that how that microstructure could be utilized in microfluidic platform to capture single cells, then pairing with 2 cells 1-1 pair, then you fuse. This is very beautiful structures, means designing with respect of design and that flow behavior of that cells in that microfluidic chip. So, here principle is that you have 2-layer microfluidic device fabricated by 2-step lithography. This is your support pillar and this is a capture cup. If you look for the capture cup, this is a 2-sided, one is front side, one is back side. In the front side there is a notch of diameter around 10 micrometer, in the back side cup diameter is around 20 micrometer, and that support pillar will help to fluid flow, and that cell will be captured first in the front cup, then it will be flowed in such a way that that will be going back to that, means from this place to that front, that larger cup, then again you give it. So, this is demonstrated in that. Okay. So, this is that configuration of that cups, and this is the distance, means if these 2 cups are that aligned around 2 micrometer spacing and 50 micrometer row spacing, that is the most desired cup alignment in the microfluidic platform, which allows around 70% of the capture that cells. Now, how cells are capturing? First, that one type of cell like say myeloma cells, we are flowing in that direction at the rate of 10 to 15 microliter per minute, then cells will be captured in that small cup of diameter is around 10 micrometer. So, 1 cells is captured. Then we are giving opposite direction flow around 100 microliter per minute, by which this captured 1 cells, say myeloma cells, will be transported to the big well of that just opposite microstructure. So, that myeloma is captured. Then you give the B cells in the same directions, then B cell will be entering in that well, like say B cells are entering, then 1 capture will be there. This is a very idealistic picture, but in a real situation what we observed that there are some clusters are there, even 3 cells are coming together, but their percentage is less. So, in that way around 70 to 90% cells will be captured just one pair. So, next is how to fuse that cell using that same platform. That could be done using polyethylene glycol, fusion efficiency around 40%, and if we give brief DC electric field for a 1 second or 2 seconds, then fusion efficiency is 78%. And both the things could be done in same platform, because PEG is very easy to give, because we can quickly flush out that used PEG using that flow behavior in that microfluidic platform, and this electric field in the beneath the electrodes are there, you can give the direct electric field. And this is a picture to showing that which are the hybridoma cells are survived and which are not survived. Which are survived, there is a clear cut demarcation of the periphery, whereas that dead hybridoma cells, there is no clear cut demarcation, and they are dead hybridoma cells basically. Now, next step is you have to capture that hybridoma cells which are fused inside that microfluidic chamber. For that we have to use that micromanipulator. That operation is like that, when you are constructing that PDMS device, that this is the cover slip, this is PDMS membrane, this is the PDMS device or the microstructures is there. Then this PDMS membrane irreversibly bonding with upper PDMS chamber, where the inlet outlets are there. Then the whole the structures is reversibly bonding with the glass plate. Then you introduced all the cells and everything what you have discussed right now. Then if you want, after that fusion, you can introduce very low melting agarose for the gel setting, to by which that fused cell will not be that much disturbed. Then you invert it like this position, then with the using micromanipulator sequentially you can recover each and every cell, and you collect in the microfluidic that micro-oil platform and let it grow. So, like say this is one example in the micro-oil, this is a blow up picture, this is the one cell and you are growing for according to the day, here we are saying that 14 days your growth, then again it will grow 23 days, which are viable hybridoma cell. It does not indicate that viable hybridoma cells will produce that antibody. So, you have to again go for that selection of the antibody selection, means how to screen that so many hybridoma cells for that antibody screening. So, this also could be adopted in microfluidic platform. So, if we look for that cell pairing efficiency of these 3 approaches or 4 approaches, non-microfluidic means macro scale cell culture, this is 5%, chemical way around say 15%, dielectrophoresis way around 20%, microstructure around 60 to 70%. So, as I mentioned that next step will be screening of that hybridoma cells for that antibody secretion, means positive clone. So, before going to that experiment, you have to select how to screen that antibody, how to means experimentally observe that antibody is secreted and assay that antibody. Here the technique is like direct ELISA, what we have done in earlier, that is macro scale process. Here principle is that say this is the secreted antibody, this is blue in color, and we have that captured antibody, this is red color, which is against that Fc portion of that antibody ligated with that microbead. Then detection antibody, which is raised against that FAV part of that secreted antibody in goat labeled with fluorescence. So, principle is that when that secreted antibody from that hybridoma cells, they will react with that first that your detection antibody, then that will be captured by that bead containing high density capturing antibody, then there will be a glow of fluorescence around that microbead. So, that is the basic principle of detection. Now, how will you screen it? Here screening is done by droplet based fluidics approach. In the this is the Y channel, this is the one channel, that bead suspension what we discussed right now is given, and another channel you are giving that hybridoma mixture of is positive clone and negative clones. Then you are flowing that to aqueous solution and using oil phase, fluorinated silicon oil phase, that using flow focusing method, pinching that aqueous part in a droplets. So, these droplets are in such a size that you can capture the only one cell along with beads. So, there is a probability of 3 types of beads, 3 types of means capture tubes, sorry, not capture tubes, is 3 types of oil phase encapsulated. This one is only beads and some your detecting antibody, and one is which captures that antibody secreting hybridoma cells. Another class will be a non-secreting, that 3 class of droplets will be there. So, after that passing that droplet, we capture the droplets in Eppendorf tubes and let it stay for another 1 or 2 hours, maybe 3 to 4 hours to secrete that antibody from the hybridoma cells. The next step will be screening that antibody secreting hybridoma cells, means which are the positive clone and negative clone. As we are incubating in CO2 incubator for certain time, antibody will be produced, and that antibody will react with that your detection antibody. Eventually, it will be captured by that beads and fluorescence will be concentrated in the beads. So, those will be screened by using laser slit and dielectrophoretic method, by which that positive clones, means which are secreting antibody, which are fluorescence positive, bright fluorescence positive, they will be captured and sorted in the droplets, and which are not producing antibody, they will be wasted. So, this is the in details of that your droplet generation and cell encapsulation process. This is 2 and 3, these are the aqueous compartment, which one compartment containing that your mixture of beads and that fluorescence antibody, then another compartment containing that all the hybridoma cells. And this compartment 1 contain the oil phase, fluorinated silicon oil phase, and when these 2 are coming, 2 phases are coming at that focus at this point, then oil phase pinch that aqueous phase, make the droplets of the required size, and size of the droplet depending upon that what speed of that all the 3 solutions, and it should be standardized, by which the droplet size will be such a that it can encapsulate at least one cell. Then this is the capture where the cells are captured, and this is the flow focusing happening here, by which droplets are generated. So, these are details of that screening. So, next step means when that cells are stored here after 2 or 3 to 4 hours according to the need, let it allow the antibody secretion, and some droplets will be glowing, some droplets, majority droplets will not be glowing. And same way again flow focusing is done it in that place, let it pass through that your screening chamber laser and dielectrophoresis setup, and this is the cells are sorted, and which are the not fluorescent, they are the going out. So, in that way we can screen around 1 million of cells in a microfluidic operation 2 to 6 hours. So, now if you can compare that 2 system, means macro scale and your fluidic system, we have a lot of leverage in micro scale system. After selecting these positive clones, to assure that these are the positive clones, the sorted droplets may be individually trapped in a microfluidic chip here, that microfluidic chip, and it will be stained to target specific hybridoma cells with the different dyes. Otherwise, you can do PCR cycle for that FAB part of that antibody. I can look for that whether it is a positive selected. So, we can save the time as well as monotonous process for the screening by it takes might be 2 to 3 months, if you are lucky enough maybe end up with 2 or 3 clones of that monoclonal antibody secreting hybridoma. So, if you can compare that throughput, cost and automation in each of the compartment, microfluidic has a higher leverage than conventional method. So, nowadays people are using that molecular biology technique by which we can screen that monoclonal antibody secreting cells, fetching that your sequence of that FAB part, then clone it, then using fast display technology and using that microfluidic technology, large scale screening could be done with a very short period of time, that I am not discussing here. But anyway, microfluidic technology has a better impact where the screening process and selection process is preferred rather than normal macro scale process. So, I think with these 10 lectures I shall be able to some idea about that microfluidic cell culture system and some readouts which are not possible in macro scale system, by which we could be inspired to adopt microfluidic cell culture in our day to day cell culture practices for 2 reasons. One, for discovery of new readouts which are not possible in macro scale cell culture system. Another thing, application aspects which has a huge impact in our modern biology. Thank you.